Hello, everyone, once again, and today I will be talking about enzyme and its function. So, what exactly is an enzyme? As discussed in the macromolecule video, an enzyme is essentially a protein that has catalytic activity, and what that means is that these proteins can bind to a certain molecule and and e- transfer it into a desired product for the cells. And these enzymes can have many more functions in the cell. An example are enzymes that can perform replication, transcription, translation, and many other functions. And if you take a look at the picture to the right, the, um, this is what an enzyme would do. And so an enzyme would bind to a cofactor, which will be discussed. We will discuss shortly. Once a co-activator binds to the enzyme, then the ligands can now bind to um, for in order for this enzymatic protein to exercise its function. And speaking of cofactor, these are molecules that assist in the substrate binding process of the enzyme and also its catalysis. And cofactors can comes in the form of metal ions like magnesium, zinc, or iron, which all exist in within the cell, and also coenzymes such as NAD plus and FAD plus. And so once an enzyme binds to a substrate, we will oftentimes call this an enzyme substrate complex. And there are essentially uh, two types of enzyme substrate complex. The first one is the lock and key model, where the substrate binds perfectly to an enzyme's binding site, as we see here. The square molecule binds to binds perfectly to the square-ish binding site, and the triangular molecule binds to the triangle site. And it's going to undergo catalysis and to be turned into products, as we see here. So that is the lock and key model. The second model is an induced fit model, where similarly we have a molecule binding to an enzyme, but as you can see, it's the shape does not resemble somewhat of the binding site here, which is where the active site occurs and where the enzyme exercise its catalytic function. So what will essentially happen is that when the substrate binds to the active site here, it induces a fit um, for the enzyme to fully bind um, onto the substrate to form this complex. And from there, similarly, it also undergoes a catalytic process to be converted into a product. So within the enzyme kinetics, now that we have gone over the complexes, the enzyme substrate complexes, I want to touch base on different variables we use to measure the kinetics of these enzymes. So the first variable that we're going to discuss is Vmax, which is the maximum velocity where all enzymes in a solution are substrate bound. Half of Vmax is what is a point where 50% of your enzyme concentration are substrate bound. And half of Vmax can be measured through Km, which is a concentration of substrate that is needed to reach half of Vmax. And S bracket is the substrate concentration used um, in these to form these enzyme substrate complex. And these K values here, the first one being K minus 1 is the dissociation constant. So what that essentially means is that even if these enzyme binds to the substrate, there is a probability that the substrate is going to be dissociated from the enzyme, which gives us this dissociation constant. Um, However, that is the opposite of K1, which is the binding constant, where the enzyme actually binds to the substrate. And K2, we often call it as Kcat, is the constant for the to quantify the catalytic process or catalysis during the um, enzyme substrate complex to be turned into products. In addition, there are also external factors that affect this um, process. The first one being pH, where there are different enzymes that works best at different temperatures. And the second one, or second more notable one, being temperature. While high temperature could increase an enzyme's activity, which means this process can happen and finish a lot faster than normal if it were to be conducted at normal temperature. Extreme temperature, however, especially with really extremely high temperature, can cause enzyme denaturation. This is where these enzymes will be unfolded and loses its function. And so these variables are placed into what is now known as the Michaelis-Menten equation. And there are also some other equations that are also used to study the enzyme kinetics. The first one is to calculate um, v, the velocity of a enzyme solution at a certain point in time. We will use Vmax times the concentration of substrate at that point in time divided by Km plus the concentration of substrate, the same concentration of substrate. Similarly, for with Km, we can use the uh, constants being discussed, the dissociation, binding, and catalytic constant to find the concentration needed to reach half a Vmax of a particular enzyme. And to do that, the Km is calculated through K2, which is the catalytic constant, plus K minus 1, the dissociation constant, over K1, the binding constant. In addition, another equation can be used to find Vmax, um, and this is rooted within the enzyme's property itself. It's dependent on the enzyme's uh, catalytic constant and also how much of a constant how much of the enzyme is concentrated in a particular solution. And so Vmax that can be found by multiplying the kcat of a enzyme with the with its initial concentration in a solution. Vmax and Km is also used in a plot called the Lineweaver-Burk plot. But in this case, this is the logarithmic function of both Vmax and Km, where the x-intercept on this plot here will be depicted be a measurement of Km in the form of -1 over Km. And the y-intercept will be measuring Vmax in the form of 1 over Vmax. And so this in order to find the slope of a enzyme kinetic of a particular enzyme, this slope would be Km over Vmax and depicted as 1b and 1s. The intercepts will be measuring the Km and Vmax, while other while other measurements will be measuring the uh, change in velocity and substrate of starting from those intercepts. And essentially, this plot is important as when we talk about it in the scope of enzymatic inhibition. The, um, the two common types of inhibition that we will often see are competitive and non-competitive. So, firstly, competitive is when a where an inhibitor, uh, like such as here, will bind the active site where the enzyme are exercise where the enzyme are performing its enzymatic function, and it's essentially competing with the substrate for that particular binding site. This will require you to use uh, more concentration of substrate to help it outcompete these inhibitors. And what you will see is an increase in Km. Um, even though we are increasing in Km, we are not affecting enzyme's property itself. So we will see Vmax being unchanged. In contrast, when we're looking at non-competitive inhibitors, these inhibitors will be binding at other sites far away from the active site, as we see here. Instead of binding towards the front where the active site is located, is binding in some other places. And this will cause a conformational change in the enzyme where the substrate can now no longer bind to this enzyme to be catalyzed into products. And since this binding changes the conformation of enzyme or one of its properties, we will see a decrease in Vmax. And however, we won't see a Km change. And essentially, enzyme kinetics and the use of enzyme is very important and is used widely throughout many different tasks. And one such application, especially in biological labs, is the use uh, of polymerase chain reaction, which is often known short as PCR, where we are amplifying a particular um, sequence of interest for further studies. Enzymes can also be used as in the form of restriction enzymes, where we are able to essentially cut DNA sequences at a particular site that is true to uh, designated for, or rather designated for the enzymes to cut. And so that's it for this video, and I will be seeing you in the next one.